In the past few decades, a new, more nuanced picture of Hebbian plasticity has emerged. In particular, scientists have recognized that for many synapses, precise timing of the pre- and postsynaptic spikes at a millisecond level matters. This phenomenon has been dubbed spike timing dependent plasticity, or STDP. In STDP, what fires together still wires together, and the neurons that are out of sync lose their link, but the definitions of these phenomena can be quite precise. The scientists Yang Dan and Muming Pu carefully measured the timing and effects of paired pre- and postsynaptic spikes in the early 1990s, and they found a remarkably regular relationship at the millisecond timescale. Presynaptic spikes arriving just a few tens of milliseconds before the postsynaptic spike result in potentiation, while presynaptic spikes arriving just a few milliseconds after postsynaptic spike result in depression of the synapse. If we plot the relationship between the relative spike time on the x-axis and potentiation or depression on the y-axis, we see a graph that looks like this. Optimal potentiation is observed when the presynaptic spike just precedes the postsynaptic spike, and strong depression results when the spikes occur in the opposite order. Why would nature have evolved such a system? A compelling explanation is that STDP exists to reinforce causal firing relationships. If a presynaptic neuron fires before a postsynaptic one, then the presynaptic spike could have plausibly caused the postsynaptic neuron to fire. If the order is reversed, then it's basically impossible for that presynaptic spike to have caused the postsynaptic one to have fired. It's not like spikes can travel backward in time, after all. This is important since many neurons exhibit a fair amount of random low-level firing, even in the absence of stimulation, and we wouldn't want to reinforce connections just because two neurons happen to fire close together in time to one another. Actually, if we look carefully at Hebb's original statement of his postulate, we even see some foreshadowing of this idea. Hebb spoke of a neuron A persistently taking part in firing a neuron B. This implied causality of a neuron A driving neuron B is exactly what spike timing dependent plasticity implements. How is this precise timing of STDP implemented in a neuron? There's some debate still, and a great deal of ongoing research into how STDP works, particularly in the case of long-term depression. But for long-term potentiation, there's an interesting idea that the backpropagating action potentials we encountered earlier in this unit play a key role. The NMDA channel is exquisitely sensitive to membrane voltage, and as we learned earlier, action potentials generated at the axon initial segment aren't confined to the axon, but also passively propagate back into the dendritic arbor. This backpropagation takes a characteristic time, dependent on the geometry of the cell, and membrane properties like the membrane capacitance. Depending on the geometry of the cell, it's not hard to imagine the wave of depolarization from a backpropagating action potential arriving at just the right time to gate NMDA receptor-dependent calcium influx. Neurons are extremely complex entities, to be sure, and we're still learning more and more about their complexity every day.